Right, guys, I figured I would just go ahead and get this done and make sure it's uploaded for you guys as soon as possible to be able to navigate and move through and begin, studying this material in preparation for the exam week. So with that being said, I want to go ahead and share my screen and make sure that you guys are able to see so that we can move through the remaining portions of the material that is in chapter 3. So again, chapters 1, 2, and 3 is what you will be liable for your first exam on Monday of next week. So we had left off then talking about, right, this concept of how we can retrieve and more importantly, how signaling takes place, right? So let's back up just a little bit. Remember, we had left in chapter 2 talking about how we generate the action potential, right? So make sure we know fully integrated, right? How does saltatory conduction happen? All of the different channels, why we have the ability to do work with an action potential, right? We set up this electrochemical gradient, we can move things and rebalance and recalibrate. So now that action potential is coming down and it's reaching, right, the terminal of my pre-synaptic cell so that it can send that electrical signal into a chemical signal with little neurotransmitters that can cross here, right, from pre to post. One way, we see that arrow, one way direction. So we said that we have calcium that comes in, right? Let's kind of walk through this process. So again, we have an action potential that comes down, and that's involved with the movement of sodium and potassium. That's not going to change. But as that action potential comes down, and again, that saltatory conduction is coming down, right? It's pushing all of that sodium. That sodium has a nice big positive charge with it, right? And as it's coming down, it's eventually going to hit these voltage gated calcium channels. Doesn't matter the voltage, right? It doesn't matter what is going to increase the voltage as long as I hit the threshold from minus 70 to minus 40, minus 50-ish, my voltage gated calcium channels are now going to open, right? And I'm going to send in a massive influx of calcium. Calcium is higher on the outside of the cell than it is on the inside of the cell. I'm using the exact same principles that I was using to generate an action potential, but now I'm switching from sodium and potassium to calcium. So now in comes my calcium. It's going to bind to my little synaptic vesicles right here, right at my loading dock, which is my active zone. And what we're going to see is that triggers the release of these neurotransmitters that are now going to cross in between this little cleft area. We call this the synaptic cleft. And it's going to bind and activate receptors on the postsynaptic cell. And we're going to start the process all over again, right? And the postsynaptic cell receiving that information, right? And it's going to make a decision. Do I send the signal down the axon or do I not? Right? So as this is happening, right, what we can see is that we need to clear and rid this because we need to get ready for the next round of release. So just like in the action potential that we have our sodium potassium pumps to calibrate, we inactivate voltage gated calcium or sodium channels, we inactivate our potassium channels, right? We hyperpolarize. And then what do we see? Our saltatory conduction comes back down in that little node to 0, right? It goes back down to resting membrane potential at minus 70. So for here, we're relying again on something to signal that we're going to do this action. So in this case, calcium coming in signals my little vesicles to release their neurotransmitter, right? But just like I need to restore the balance underneath my little node of Ranave to allow for the next action potential to occur, I need to make sure that I can recalibrate here, remove all of my neurotransmitter, so that when the next round of release comes, it is able to activate the postsynaptic receptor again. So again, if I don't remove my sodium and restore my potassium sodium balance, my battery dies, right? I can't regenerate any more work. My paddle wheel stops spinning. In this case, if I don't clear out my neurotransmitter, then I've just got a bunch of chemicals that are hanging around that are constantly activating these postsynaptic receptors, and I'm not creating my battery, right? It just sort of dies out. Now, we'll talk about this extensively inside of things like different types of disease states and different types of addiction states and with different drugs and how this is worked. For the purposes now, all I want you to remember is these series of steps that are action potential that we were just talking about in chapter 2 is a electrical signal, and it now transmits into a chemical signal, right? And that this switches, right? So let's go back and look at this really quick. Oh, so again, as we can see out down here at my synaptic cleft, at my terminal, right? This little foot that looks like a little foot, right? Notice here I don't have any more myelin. I don't have to worry about it, which is why this interaction between our calcium channels and things to come that we are going to talk about, including receptors on the postsynaptic side, right? They can now play with the external and internal environment. It's only underneath the myelin, once I have the wrapping of the wire, that I can't exchange ions, I can't exchange with my external environment. So that's why we have to have the little breaks at the node of Ranvier. Once I get down here, notice my myelin is no longer there. My myelin has been removed so that I can play with the external environment, and I can just allow for things to easily move across the membrane. So just things to remember. We had left again by saying that there are certain types of drugs that can inhibit this process of chemical release, of neurotransmitter release. And that can be through things like our botulinum toxin, right? It comes again from this bacteria that, you know, emits out or secretes out this particular toxin. And this toxin cleaves, cuts off the ability for neurotransmitters to now be released, right? And so again, what would I ask you? I just would ask you, you know, give me an example of something that would inhibit release. You know, maybe I could ask you, what are other uses of Botox besides, you know, cosmetic? Here are some of these examples, etc. So as you can see, my little synaptic vesicle is made of membrane, right? And as it's coming down to dock here, right, it physically adds to the membrane. That's what I mean, where I can't keep growing and growing and growing. There's very limited space inside of the brain. So with that being said, I know that I'm going to need new vesicles to refill with new neurotransmitters so that my neurotransmitter release can continue to occur. So as we add this little vesicle and it fuses and becomes flush with the membrane, right? Because it's fusing and it's dumping its contents out. There's a little conveyor belt on the other side known as the process of endo within, right? That's pulling that membrane back up, and it does exactly that. It just pops off a little vesicle, we reload that vesicle, round and around and around and around, right? We call this the synaptic vesicle cycling process. This is the cycle of exo to endo, exo to endo, exo to endo. Neurotransmission to making new, neurotransmission to making new. And so again, we are going to get into the deep dive of individual types of neurotransmitters. But for now, what are we doing? We're just setting up this concept of, again, what do neurotransmitters do in the body? What are the different types? what do I have to consider? How do I classify a neurotransmitter? And so again, there are those that we would put into the bucket of being called sort of the classic neurotransmitters. These are the things that we are going to spend the most amount of time talking about. These are those that are stored in the vesicles. They come down and dock. They release those neurotransmitters. They pull up a new vesicle. They reload, and around we go. We also can see that we have other things that can slip across the membrane, known as gaseous, or lipid transmitters. So these are things that are synthesized on demand, and they act as retrograde. This is going to be something that we're going to look at extensively when we get to marijuana. You have endocannabinoids, endo, within, right? Naturally occurring. And people may take exogenous marijuana, THC, CBD. But what that does is it actually activates receptors and transmitters in the postsynaptic cell that are actually sent back to the presynaptic cell. So wait, Dr. Varga, I thought you said that it's only one-way direction. It is. The action potential is always only going to come down the axon, hence the name action potential, and hit that terminal and again, either fire or not fire, right? Send the neurotransmitter out or don't send the neurotransmitter, right? Electrical to chemical, pre to post. That is going to be the case. But after pre to post happens, activation of that postsynaptic cell by releasing of the presynaptic cell sometimes will send messages back. It's not an action potential because it's only happening here at the terminal. This we call retro, right? Just like we saw in moving things. Let me see. Right? Antrograde and retrograde, right? We saw this kind of when we send things down the axon and then bring them back, right? It's the same concept. It's we have antrograde, towards, and retrograde, back. We'll talk about this extensively, but for the most part, we're going to focus more on our classical as part of that one-way trajectory from pre post. All right, so now I have defined how I turn an electrical signal into a chemical one. I have defined how or what neurotransmitters may look like as far as kind of the base requirements, right? We talked a little bit about the different types, the different major categories. We haven't talked about anything specific yet as far as, serotonin or dopamine or anything. We've just given major categories. We talked a little bit about fast versus slow. Again, depending on what I'm making and where it has to go from and to, in the case of the soma down to the terminal. It's going to take time. But now I need to know how or what is going to affect how often those neurotransmitters are going to be released. Well, if we guessed how often the action potential is firing, that's absolutely one way, right? Self-firing rate. How often am I receiving that signal down the axon to the terminal to say release, release, release? There also is this thing, you do not have to go deep down this rabbit hole, but again, there is this concept that we call about the probability of release. How many vesicles are in the active zone ready to go? How much neurotransmitter is inside of those vesicles? How much calcium is available to the system, right? All of those things fall under that biological swing that we have to add into this concept of probability of release. And when we start talking about these different drugs, this is going to become really important when we talk about biological swing, because not everybody has the same amount of neurotransmitters, the same amount of receptors, right? Or the same level of calcium. And these things all can affect that probability of outcome. Lastly, we can also look at things like autoreceptors. So autoreceptor self, we actually can see that there is times when the neuron itself can auto-regulate itself. So we're going to talk about this a little bit more, but let's look at some different pictures and images of what I mean. So for an autoreceptor that is on the terminal end, right? The synaptic terminal. An autoreceptor that is placed down at the terminal, because again, just think about this. You have a brake pedal in the car, you have an emergency brake, you have emergency lights, right? You have a lot of things that are available to let people know things aren't going so great if they're not going so great, right? You have a horn, or you just straight up panic. But if there is a mechanism that needs to shut down release, too much firing, right? Again, maybe I'm getting a postsynaptic signal that's coming back, or maybe I'm sending a self signal based upon how much neurotransmitters are being released. If they can't be cleared, what happens? What do we see? We see that that neurotransmitter will build up inside of the synaptic cleft, and it's just a matter of concentration gradient again. This is too much. I can't clear it in enough time. So we put these terminal autoreceptors further away from the active zone as a fail-safe, as like the bumpers on the bumper car. Sure, I want to release at my active zone. I want to have my docking site ready to go. If I'm releasing under normal conditions and everything's doing its job, that's great. My autoreceptor shouldn't theoretically be on. But maybe I have the case of too much self-firing is happening. And now I'm releasing so much neurotransmitter that's moving away from the active zone, and now it's spreading, it's creeping out beyond this little cleft, the pre and post, such that it's meeting and kind of wrapping around. I'm sort of just flooded with neurotransmitter. This autoreceptor will be pinged, will receive that neurotransmitter that just came from the neuron it's attached to, and say, uh-oh, right? It's like the water in the bathtub is now overflowing. I shouldn't be sending out anymore. I can't even clear what's currently there, right? So this terminal autoreceptor basically comes in and it says, don't release anymore. Let me try and see if I can drain some of this bath water out. We also can have, again, that capability of somatodendritic. So again, a dendrite can synapse directly onto a soma, the big mainframe house of the cell, and say, let's just slow down a little bit. Let's inhibit. So again, this receptor, once it's in that postsynaptic cell, now it's self. Because remember, our signal sends from the soma down the axon to reach the terminal, to switch from electrical to chemical, from pre, post, and send the signal onward. If I'm receiving information from all of my little dendrites that are saying fire, 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 right? Keep going, keep sending those action potentials, keep doing it. Think of this somatic dendritic autoreceptor, again, it is on the soma of the receiving cell, that it's basically saying there is too much going. We need to shut down. Quit sending those action potentials. It's just another mechanism to make sure release is able to clear, rid, and be ready for the next round, right? We have to get back to baseline in order for the release to do its job. It's just like taking the drug, right? If I, or anything, think about alcoholism. If I continue to drink every single day, that tolerance is going to go up, that threshold is going to go up. So as I mentioned, what if I can't break down? Well, let's look at the ways in which we need to clear and rid, right? My synaptic cleft, wash out that bathtub so that we can refill it again, right? The first thing we have is that inside of the synaptic cleft, mainly located, it can be located pretty much in a lot of different places, sometimes on the pre, sometimes on the post, but for our purposes, let's just focus on the post based on this picture here. There are little enzyme molecules or precursors that are ready to go. Just like enzymes can put things together, enzymes can break things apart. So if I want to refill the bathtub or clear out my synaptic plaque so that a new round of neurotransmitter release can cause the effect in a postsynaptic cell, I want to break those down. So those enzymes will be secreted out, they'll gobble up and break down all that neurotransmitter, it'll clear and reduce the concentration of neurotransmitter in the plaque. I can also vacuum cleaner, as I like to call it, through transporters. Notice this is not an autoreceptor. This is what we call a transporter. This transporter is saying, I'd like to reuse, reduce, and recycle. I don't need to make and package and redo new neurotransmitter. Let's just suck up what was released and see if we can't repackage it to reuse it again, right? It's like those chargeable batteries. Once they go dead, I might as well just plug them in, recharge them back up, and I can use them again. Remember, I also said that in the comparison of glia cells to neuronal cells, there's about 3 times as many of our glia cells, and we do a lot of things, including, right, taking those nutrients that are kind of coming in from the blood-brain barrier and now cross over into the glia cells, and the glia cells sort of distribute this. They also can be responsible for helping to clear that synaptic plaque. They're always sort of hanging around as sort of the referee. What's going on? Let me make sure. So again, 3 different ways. Enzymes, vacuum cleaners to suck up any extra and repackage them inside of the presynaptic terminal, or I can have my glia cells take them up via transporters as well. So why is all this important? Because as we continue to move through and look at different neurotransmitters, they have effects across the body. Not only are we going to look at things that are inside of the central nervous system, but we have a whole nervous system that's outside the peripheral nervous system. So as far as you are what you eat is concerned, that's very true, because what you have is what we now call the gut-brain axis and neurotransmitter release. In fact, many of the neurotransmitters, serotonin being one of the biggest, is actually produced by the gut bacteria that you feed through the types of food you eat. So again, if you're feeding your body good, healthy, nutritious, good things, you're keeping stress low, right? Back to that environmental thing, you can allow for your gut bacteria to be in that perfect biological range that's great for you to thrive and survive, right? And they're going to help to create and generate neurotransmitters for you. Those neurotransmitters are used by the body. So drugs can absolutely alter this, especially if you're looking at things like antibiotics. Antibiotics, what do we know antibiotics do? Well, we know that they kill off bacteria. They may kill off the bad bacteria, but unfortunately, they're also going to kill off the good bacteria inside of your gut. This is why sometimes they say, yogurt, or make sure that you're getting a pro or prebiotic, right? Or eating good, healthy prebiotic foods, your leafy greens, get some good fiber in there, feed your good bacteria again so they can restore your microbiome, right? And this microbiome that feeds you is absolutely feeding back to the bloodstream and giving you all of the good things that it's done for you by breaking down your food. So now, again, we call this the gut-brain axis. And this, of course, refers to that signaling cascade between how the gut influences what the brain is doing, which influences what the brain is, right? And vice versa, around and around we go. These things are very important. This is a huge shift that we're seeing in neuroscience. And there's lots of research that's out there on this. And again, one of your discussion board posts is looking at this. Okay, I am going to pause here. I'm gonna come back up and wrap out the remaining portion of these slides in the next one, just so you have 20 minute, 25 minute chunks of digestible material.